That's great, Anne. Thank you. It's really exciting to hear sort of your experiences in big pharma and also, in smaller working groups. So I'm a human oncologist, but I've had a longstanding interest in comparative oncology. When my first job outside of my fellowship in Seattle was actually working at Pfizer, where I was launched into being the global clinical lead of our hedgehog program. And I immediately recognized that it was going to be very, very hard for the first in-human study to gain traction. And as Anne really nicely pointed out, there's a lot of internal competition for resources in large pharma, particularly in phase 1. We've got an embarrassment of riches in terms of molecules to bring forward. And so those of us clinicians that are leading these big phase 1 teams are always looking for ways that we can sort of get to the top of that funding barrel. And so the bar is really high to do that. And one of the things that I did very, very early on was I brought Chand in to talk about how to demonstrate, reasonable clinical PK and PD in a companion dog model to sort of augment our clinical program. And so with that, I'm not gonna spend too much time here because we've talked about this a lot. There's an opportunity and there's a problem and there's an opportunity. And so what I really wanted to just focus on here just for a couple minutes in light of some of the other speakers is that again, the success rate for bringing oncology drugs forward successfully into phase 3 trials is really dismal. And so the goal for us in drug development is to kill early. So that's our goal. If something isn't active in this era of personalized medicine in phase 1, we're usually in deep doo-doo in terms of being able to bring that forward. And so that's just the fact of the matter. But the issue is in personalized medicine, we've learned that there really is no MTD for a lot of these targeted therapies. So this idea of bringing this 3+3 design and sort of this, dogmatic approach really doesn't work in phase 1. And so for many of these molecules, because there isn't an MTD, we're forced to be a little more creative and figure out what others have called today the biologically active dose, for example. And so we've learned that more isn't better for targeted therapies. And I think that was actually shown very nicely in a meta-analysis from MD Anderson several years ago. And in fact, when you actually dosed up with these targeted therapies, there was potentially more toxicity and less efficacy. And so that's really, again, brought us back to how can we design early phase clinical trials much more smartly. The PK/PD model is really where it's all about. And so it's all about trying to identify a validated and qualified biomarker early on in these trials. And if we can determine that in a preclinical or companion dog model and bring that forward hand in hand, that can really tend to jumpstart our early development and also add data so that our investigators are actually enthusiastic about enrolling on this trial. So it's really an iterative process. It's bringing sort of going from the bench to the clinic to the bench and back again as we learn more and more. And so hopefully this process will be not just in early development, but will tend to be iterative because, again, more data involves, that this is what gets investigators excited. Finally, another point that was brought up earlier by Dr. Hellman, I think yesterday, is the problem of endpoints. And this is really important, particularly for hematology trials. So there's been a very recognized disconnect between a response rate and survival. And so, we started to sort of see this in solid tumor trials, and it's really become clear in hematology trials. The drug Mylotarg actually was initially approved for a very robust response rate in acute myelogenous leukemia and subsequently was pulled from the market because the survival rate didn't correlate with that. So that's a big problem. That's a big hole in drug development. So anything we can do to understand that better by working in the companion dog field would be a huge win because that's something that the FDA is very, very interested in. Certainly we as drug developers are interested in both the canine and the human space. And then finally, again, just to come back to enrollment, it really is data-driven, and all of this really, really helps us to enroll these trials, particularly in the world of having a pediatric investigative plan for every molecule that's going forward as well. So with that, that's really all I wanted to say, and I'll join the next speaker, and I think I'll join the panel a bit.